Hello students, in this video lecture we are going to learn about the viral disease, that is the tobacco mosaic disease, right? It's a very common and famous disease, right? That is the tobacco mosaic disease. So we will learn in this video about this disease in details, like the host plant as well as the causal organism, means pathogen, what are the control measures of this, so that we are going to learn in this video lecture, right? So here you can see this is the tobacco plant, right? So in our state, in our Maharashtra, there is no authority to cultivate the tobacco plants, right? But the states like the Karnataka, right, as well as some state like the Bihar, right? So on such state they have the authority to cultivate the tobacco plants, but in the Maharashtra not allowed, right? So that's why I'm showing this photograph to you. So how is the plant of tobacco, right? So this is the field of tobacco. So let's start with the significance of the disease, why we have to study this disease, right? The first recognized virus disease of the plant in the world is a tobacco mosaic disease, right? So this was the very first disease in the world which has been noted as a viral disease, right? So most common disease, it's on the tobacco plant, right? In the India, particularly 13 states like the Andhra Pradesh, Assam. Here you will get the Maharashtra, but in the Maharashtra there will be no authority to cultivate the tobacco, so just remove it. But in some agriculture field there might be a possibility to cultivate tobacco plant by illegally, right? No, but according to my knowledge there is no any legal authority to cultivate this plant, right? So these are the different kinds of states where the disease get found. The first virus where the protein coat was fully sequenced into 158 amino acids. So it's proteins, right? So protein is sequenced by the scientist, and this is the very first virus which have been its protein coat was fully sequenced, right? The tobacco mosaic virus was the first plant RNA virus of which the complete genome was sequenced and to which the monoclonal antibodies were produced. So there are some varieties of tobacco, right, which has been adapted for this virus, because we have all the knowledge of this virus, because its genome, that is the RNA, it's fully gets sequenced, and as we know the all details of this virus regarding its genome, we have developed the monoclonal antibodies, and thus some plants they are adapted for these viruses. What are the economic loss caused by this tobacco mosaic virus? So it reduces the quality as well as the quantity by up to 50% and up to 30% worldwide respectively, right? So the quality it reduces by 50% and the quantity of the production it gets reduced by 30% in all over the world, right? So it will currently 1% of the yield loss in the all over the world, because the resistant tobacco varieties are routinely growing. So that's why the economic loss is nowadays it is going to be decreasing, right, up to the 1%. It has a wide range of host. So this tobacco mosaic virus, so first found in the tobacco, that's why the name is given to the virus is a tobacco mosaic virus, but it can infect another plant also, like the papaya also, right? So this virus has the wide range of host. It means it is not host specific, not restricted to one particular host. It can infect different kinds of host plants. It causes the 20% of yield loss in the tomato also, right? So as it is not attack only on the tobacco but also cause the disease on the tomato, and thus it causes the 20% of yield loss, right, in all over the world. So tobacco mosaic virus has been known to cause a production loss for the flea cured tobacco of up to the 2% in the North Carolina in the United States, right? So this is about the economic loss. Then we will discuss about the distribution, right? So India is the second largest producer of tobacco with the annual production of 800 million kg, right? So we are famous for bad things also, right? So India, it generally it's a large producer of the tobacco, right? As some states, some South Indian states, as well as the North Indian states, as well as the Seven Sister, you know the Seven Sister, right? The northern east part of the India. So there also they can cultivate the tobacco, okay. But in Maharashtra, as well as in the uh, Gujarat also, the cultivation of tobacco it's strictly banned, right? So it is worldwide in distribution. Means what is the meaning of that? The, it is cosmopolitan everywhere in the world, like in the cold temperature or hot temperature or moderate temperature geographical region, everywhere it's get found, right? Up from the United States to the India, Sri Lanka, as well as then United Kingdom also, UK also, Japan also, Australia also, everywhere it gets spread everywhere, right? So over 20 different virus disease occur in the tobacco, in which the mosaic disease is the most important disease. So tobacco faces different kinds of viral diseases, up to 20, but one of the most severe, the most important disease is the tobacco mosaic virus disease, right? The host plant, obviously, it's a tobacco plant. It is known to infect the member of different 9 plant families of 125 individual species, right? So this is the wide variety of these viruses. It including the tobacco, tomato, pepper, means all the member of Solanaceae family, like the brinjal also, okay, uh, uh, potato also, then the cucumbers, and a number of ornamental flowers, right, like the lily also, okay, gerbera. So they can infect to such kind of different ornamental plant, flowering plant also. History: the Adolf Mayer, it began to study the tobacco mosaic virus in 1886, right, for the first time. In 1898, the Martinus William Bijerink, he proposed that the tobacco mosaic virus was caused by an infectious fluid, right, which is he called as a virus, okay. So what this scientist said, it says that the tobacco mosaic virus was caused by an infectious fluid, and that fluid it get name it as a virus. So in 1935, it was the first virus to be crystallized by the scientist W.H. Stanley. So this Stanley scientist, he first crystallized the virus. Crystallize means it convert that viral particle into the crystal form to study, right? So this has been done in the 1935. Then scientist Koch in 1936 took the first electron microscopic photograph of tobacco mosaic virus, right? So he took the photograph of this tobacco mosaic virus. Otherwise, no one knows about its structure. The Girer and the Scram in 1956 and the Frankel and Conrad in the 1955 showed that the RNA is responsible for the infection. Right? So what they have told, RNA is responsible for infection. It means, what is the meaning of this? This virus, that is tobacco mosaic virus, having RNA as a genetic material. So in 1969, the scientist Takebe used the tobacco mosaic virus for the infection of suspended tobacco leaf protoplasm or protoplast. So thereby providing the basis for the synchronous infection system for the studying the virus replication. Right? So this is the history of this virus. Then we will see the symptoms. So how this tobacco mosaic virus caused the disease on the tobacco plant, and what the tobacco plant show the symptoms in response to the infection of this virus, infection of the tobacco mosaic virus. The light discoloration along the veins of the youngest leaves. Right? What he shows the symptoms? Light discoloration. Discoloration means the degeneration of green color. So this degeneration of green color along the veins of the youngest leaves has been shown by the house plant. The latter, it turn into the dark green pattern associated with the veins. It means whatever the veins are present in the leaves, right? So they will turn out into the dark green color. So this is the pattern of association regarding the veins. The severe infection causes the narrow, puckered and the thin leaves. So leaves you have seen the normal leaves, but if the virus will infect the leaves, then what will happen? The leaves will remains narrow, puckered. Puckered means they show some kind of coiling, and thin leaves, right? Then the growth of the plant remains stunted. The mosaic pattern of the light and the dark green, or we can say yellow and green on the leaves, this separated by the sharp line of demarcation. Okay, so this is very important symptom of this virus. The malformation of the leaves or growing points is another prominent symptom of this virus. The distinct yellowing only of the veins, right? So leaf having the veins, right? Particularly the dicot plants having reticulate venation, so it's just prominent. So what happen? Only the veins, they will be yellow in color, and remaining part will be remains green in color. So that's why you can easily notice that yellow colored vein, right? The yellow patches, they develop on the leaves due to the presence of more than one strain produced by the mutation. As we know that RNA is unstable as compared to the DNA, right? So that's why genetic material is what? The genetic material of tobacco mosaic virus is RNA, right? So as it's having RNA as a genetic material, so its genetic material get mutated, and due to the mutation, what will happen? Its structure as well as its genome will going to change, right? Then here you can see the yellow vein caused by the virus. So here you can see the remaining part, it remains dark, but you can see the veins, midrib as well as veins. So they are yellow in color, light yellow in color, right? So you can easily distinguish that this is the infected plant by the tobacco mosaic virus. In another photograph you can see there is a dark green patches. So this patches also developed by the infection of this tobacco mosaic virus. Okay. Then we will see the systematic position or taxonomic position of this virus. So this is the virus which is unranked. The phylum is Negarne and the order is Incerticedes. The family of this virus is the Virgaviridae and the genus is Tobamovirus and species is tobacco mosaic virus. Tobacco virus and tobacco mosaic virus, right? So this is the systematic position of this virus. The causal organism is obviously it's caused by the Nicotiana virus 1, right? And its molecular weight is near about 39 million daltons. Okay, so it is rod shaped, gram positive, measuring about 280 into 15 to 18 nanometer. It means its sizes varies from 3000 angstrom unit in length and its diameter is like the 180 nanometer. Sorry, 180 angstrom unit. It means it's a rod shaped virus with a hollow tube of about 20 angstrom unit in diameter. So it's a tube like structure. It's having a tube like structure, right? And this tube is a hollow tube and the diameter of this hollow tube is 20 angstrom unit. Okay, so it is made up of centrally placed ribonucleic acid, that is the RNA molecule, which convert with a protein coat that is called capsid. Okay, so that protein coating of the virus is called as a capsid. The protein subunit, near about 94.4%, is composed of the single chain of 158 amino acid residue of known sequence. So we know the protein subunit, which is composed of this single chain peptide, that is 158 amino acid residue. The folding of the chain is ellipsoid with an axial ratio of 3:1. The RNA 5.6% is single stranded RNA. It means it will be more unstable. The molecular weight is 2.4 million daltons and the helix radius of 40 angstrom unit that can be seen under the electron microscope, right? So here you can see the actual photograph of electron microscope of this tobacco mosaic virus, right? So I will just zoom in. So here you can see the rod shaped structure of this virus. Okay, its diameter is short and length is very long, and this tube is hollow tube. Here you can see the structure of this virus. It is elongated, which is coated by the protein, right, which is called as a capsid. Whatever you can see the pupa like structure, right? So that pupa or petals like structure, it's a protein coating which is called as a capsid. Then nucleic acid, that is the RNA, it is present inside that capsid, okay, which shows the diameter of 180 angstrom unit and the length of 3000 angstrom unit, right? So then many capsid, they forms the capsomers. Okay, so this is the clear cut structure of this virus. Okay, then here also you can see the nucleic acid denoted by one number, which is looking like spiral shape. It's actually RNA. Then capsomer proteins, that is protomer, it is denoted by second number. You can see the petal like structure, and the third is the capsid. Whatever the surrounding is there, which is blue in color, so that is a capsid. Okay, so this is about the structure of this tobacco mosaic virus. Then we will see the disease cycle. The primary function, primary infection of this virus, they shows the transplants are handled by the workers whose hands are contaminated by the virus. So this during the transplantation and the handling of the workers, so sometimes what happen, some plant they will be infected by this virus, and when they are just plugging off these viruses, when they are handling such infected plant, okay, then these workers will, what they will do, they will transmit this virus as the virus is present on their hands. They enter into the host cell through the wounds or the after the wound, right? Then it entered through the wound, it replicates, spread through the plant body and causes the diseases. Okay, so it cannot be enter inside the plant body if there will be no wounds, right? So if there will be wounds only, then it can enter inside the plant body. The secondary infection, so this is the first infection. The secondary infection is what? It occurs after the transmission during the suckering of the tobacco and other cultural practice. So it continues throughout the season, resulting in the severe infection. Okay. The virus is a sap transmissible, means it's present in the sap and enters through the wound. So no insect vector is known to transmit this virus. So there is a no vector up till now found to be transferred this virus, okay, easily transmitted in the field by the mechanical means and by winds and water. So if this virus will be dissolved in the water, okay, so during the watering of the field, then it can be possibility to spread, or by wind also it can spread by wind also, okay. So the various cultural operations such as topping, clipping, and this should spread the virus from the diseased and the healthy plant. So these are the different cultural operation or cultural practices done by the farmers, like the topping, clipping, and the shoot spread. So during these cultural practices, the virus can be spread. So this is the life cycle of virus, right? So systemic spread will be developed. The mosaic symptoms, the cause on the leaves, and after that the seedlings infection through the contact. So in this way the cycle will be done. So what happen, sometimes the nursery plant, generally plant is preferred for the plantation. So during the nursery there may be a possibility of getting the infection by the seedlings. So if these seedlings are infected, then tobacco mosaic virus symptoms will be shown on the leaves, right? So this will be takes place at the off season as well as at the systemic spread also. So this is the overwinter or the over summers in the infected tobacco stalks and the leaves in the soils, right? And in this way the life cycle of the TMV, that is tobacco mosaic virus, will be completed. What are the favorable conditions for the spreading of the diseases or getting the infection? The tobacco mosaic virus is one of the heat stable, means thermostable virus. The heat inactivation point of the virus in crude juice extract from the infected plant is being 90 to 93 degrees Celsius for 10 minutes. So in such a high temperature it can be remains as it is. So it's a thermostable virus. So there will be no effect of high temperature on this virus. The virus retain its ineffectiveness, that is the stability, in the dried mosaic infected plant debris also, even at the temperature of 120 degrees Celsius for 30 minutes. And they are not completely inactivated during the process of tobacco products, right? So it means if someone is eating the tobacco, right, consuming the tobacco, then it can be enter inside its body also. But this is the plant viral disease, so it cannot affect on animals. The virus protein synthesis. How the virus protein synthesis takes place? The viral RNA itself plays the messenger RNA, right? Otherwise, the central dogma says DNA can replicate, DNA can convert into RNA, RNA can convert into proteins, right? But not RNA. All the RNA can convert into proteins. Only mRNA can convert into proteins. But this viral genetic material, that is RNA itself, act as a messenger RNA, right? So the virus utilizes the amino acids, ribosomes, and the transfer RNAs of the host, but it becomes its own blueprint, and thus the proteins get formed. The first RNA produced is not a viral RNA but a complementary strands, that is a mirror image of the virus, which is temporarily connected to the viral strands. This is the infection process of the tobacco mosaic virus, right? So tobacco mosaic particles, tobacco mosaic virus particles means when they are free in condition, the virus will remain as inactive, or it will be nothing but a dead particle. But when it enters into the plant through the wounds, then it releases the RNA strand by shedding its capsid. So capsid it will get discard. Only RNA will insert inside the host cell. And the synthesis of virus replication by the association with the ribosomes will be takes place. Then virus replicate the synthesis of complementary RNA strands, and then the formation of the strand means original genetic material of the virus will be formed, and the replication of these RNA strands will be takes place, and after that it associated with the host ribosome, and the protein coat that is the capsid will be get synthesized, and assembly of the coat proteins and RNA will be takes place, and in this way this virus will be get reproduce, and it can affect to the another plant body also. So this is the infection process of tobacco mosaic virus, right? So this is the life cycle of the viruses. Okay, so here we will start the host cells which got the viral infection. See the capital A diagram. So in the capital A diagram you can see the host cell as well as virus. So this virus just inserted inside the plant cell, and after that what will happen? It will degrade the viral protein sheath, but viral RNA will be get enter inside the nucleus. Then what will happen? The complementary or the replicative RNA synthesized by the viral RNA, and then the newly synthesized viral RNA from the complementary RNA will be takes place, and thus with the help of ribosome as well as the tRNA, the amino acid forming the polypeptide chain, and after that the protein subunit or the capsomere will be get formed. Thus the viral RNA and the capsomers they get integrated and forms the viral particle. So in this way this cycle will be get completed. Then we'll see finally the disease management, integral disease management, right? IDM. In this what we are going to learn how to control the diseases. So there is no chemical cure for the virus which is infected plant, but the sanitization and the use of resistant varieties like the CC37, NC297, Jayasri Yamar, and the Godavari by the CTRI, that is Rajmundri, etc. So these varieties are resistant to the viruses, so you can use it. The purchase the virus free plant from the tissue culture, so that will be beneficial, and the field should be bedded thoroughly, and regular rugging should be done so that this virus maybe remains in the water or somewhere. Okay, so if you are removing all the weeds as well as you are doing rugging, then the field will be clean. Okay, in the US, United States of America, the spraying of the plant with the milk before transplanting or otherwise dipping the hands in the milk during the transplanting and the handling is shows the benefits, right? The cross, as the viral particle, they may be present on the hands of the workers or farmers. Cross protection had been reported if the plant are inoculated with the milder strain first. The genetic engineering also possible in which the N gene, right, it is responsible for the resistance to the tobacco mosaic virus. The N gene is transferred into the Nicotiana tobacco plant and the plant will become genetically modified. So this N gene is transferred into Nicotiana tobacco and Nicotiana glutinosa. So, but most commonly Nicotiana tobacco get cultivated. The interaction of N gene products with the tobacco mosaic virus, a hypersensitive reaction occurs which cause the plant to kill its own cells at any point where the virus entered. Okay, so in this way the protection get means the infected cell will get died by the plant itself. Subsequently, the lesions form on any inoculated leaf, restricting the movement of the virus to other plant parts and to other healthy plants also. So here we will stop here. Thank you.